RNA sequencing profiling of these fibrous caps from patients that either had a stable plaque phenotype or an unstable ruptured phenotype. And the data is kind of summarized on the left. So we end up with having roughly 120 transcripts that survive multiple testing, so are above this kind of greenish Bonferroni cutoff line. And 14 transcripts, so these were actually these orange dots that are either to the right or to the left, that have a fold change of greater than 2. And out of these 14 transcripts, 10 were actually long non-coding RNAs, again, letting us assume that long non-coding RNAs must have a fairly important function in mechanisms that drive lesion stability. And out of these, I briefly want to mention this one because it's very interesting, or it has a very interesting expression profile. NUT6.
I wanted to ask how much. Wait, I just wanted to ask how much Bitcoin it would take to buy a fat nigger juice life.
Sorry, can we maybe keep questions? I didn't really hear that, but
how many
questions after the
Okay. I would ask the question after.
I think that's easier. I think you can put it into the chat. I think that probably works a little bit better if you write it down in the chat. I'd be happy to stay along.
I don't know how to use chat.
Hello?
Question. I think that
question is how much Bitcoin will it take to buy a fat nigger juice life?
Hello?
Okay.
Can you guys hear me? Hello? Hello?
Yes.
You fucking boomers, you're not being hacked.
Okay, maybe keep going, Lars. Lars is muted by accident. Let's unmute Lars here. Okay, everyone else keep your microphones on mute, please. Thank you.
All right. So again, this antisense transcript, this long encoding RNA was kind of interesting because so NUT6 is an antisense. It's also called FGF2 antisense because it's transcribed on the opposing antisense strand to the fibroblast growth factor 2, so FGF2. And both were actually significantly deregulated. So NUT6 was significantly higher in ruptured lesions, and FGF2, the sense partner, is significantly higher in stable plaques versus ruptured lesions. And there's a very nice interaction between these two. And Hannah Winter, a postdoc, PhD student in the Munich lab, is currently dissecting this mechanism and has shown that basically by knocking down NUT6, you can increase FGF2 expression, and FGF2 really is a growth factor. And by that, you can stimulate smooth muscle cell survival in fibrous caps, and that leads to a much more stable plaque phenotype. But the long encoding RNA I will mainly talk about today, because it's very soon going to be published, hopefully, is this long encoding RNA termed Myatt. So Myatt was also significantly higher in ruptured lesions compared to stable lesions. And we also confirmed this in this second cohort that we have access to, which is this biobank of Karolinska and atherectomies, again, together with Ulfedin and Lubica Matic. We were able to show that Myatt really is, in advanced lesions, really one of the top, if not the top, regulated target. And we could confirm this in patient specimen with a symptomatic disease. So BIKE is a little bit different. It doesn't really look at plaque morphology, but it looks at the clinical background of these patients. So whether these patients are either symptomatic or asymptomatic. And again, Myatt was very significantly elevated in patients with symptomatic carotid artery disease compared to asymptomatic patients. And then, so this is really the last piece of information from human biobanks that we have, again, together with, in collaboration with the Max Planck Institute in Martinsried. We again worked out a protocol using laser capture microdissection to cut out different areas of advanced lesions and established a protocol where you can use very fresh tissue that is specifically stained so that a machine-based system or a machine-trained system can actually identify these different areas, and we can cut them out. And as Matthias Mann's lab is mainly focused on proteomics, we've done a lot of proteomics on this, but we're also able to look at on the transcriptome level, on the RNA level, and here also see that this Myatt long non-coding RNA has or is very much enhanced and very much high expressed in fibrous caps from advanced lesions compared to the necrotic core, as well as the media of these advanced plaques. And then just to localize again where this Myatt is in human disease. So this is also kind of an interesting stain because it kind of shows you how severe atherosclerosis really is in advanced stages, in this case, again, in the carotid arteries. And so I lost my mouse, no, it's still here. So this is kind of like the old media that is still, or it's all that is basically left from the healthy vessel, and everything that is happening in between is really disease. So this is all that is left from the lumen. And Myatt, again, based on in situ hybridization, has a very interesting expression profile, again, along the fibrous cap, sitting on top of this of these huge necrotic cores, is in particular in these so-called shoulder regions where these plaques are usually very likely to rupture. And based on immunohistochemistry, we're able to show that it very much co-localizes with in SMC rich areas, so cells that also express that are SMA positive, so smooth muscle cell and alpha actin, and to a lesser extent also with CD68 positive cells, so phagocytotic cells, and most likely macrophages. And then just to establish what the function of this long encoding RNA of Myatt, we performed studies using live cell imaging where we knocked down an overexpressed Myatt, and we're able to see that if we use gap-MERs to inhibit Myatt expression, we significantly in these human carotid artery smooth muscle cells. And kind of as a gain of function study or as a confirmation study, we're able to see that the opposite happens if we do a plasmid-based overexpression of Myatt in these human carotid artery smooth muscle cells. Yeah, so how is Myatt doing this? And there's, of course, a couple of different ways to establish functionality of different long encoding RNAs. There's a couple of bioinformatic algorithms. One I want to introduce you to today that some of you might even know, which is termed long target. So long target looks at or predicts binding sites for long encoding RNAs in the promoter regions of transcription factors. And for most of the long encoding RNAs, we end up with maybe 10, maybe 12, maybe 20 different transcription factors. But in this specific case of Myatt, we really could identify only one transcription factor, one gene that Myatt binds to, and that's KLF4. So the promoter region of KLF4 has a binding site for Myatt. And KLF4, of course, is a very prominent mediator in the athero field. So based on this really groundbreaking work from Gary Owens, but also others like Martin Bennett and many others, we know that really maybe the majority even of phagocytotically active cells that become foam cells and are important to lesion formation are not necessarily the monocytes who come in through the lumen, but are really smooth muscle cells who are very plastic and that can transition or transdifferentiate into at least a macrophage-like cell that becomes phagocytotically really active and is important to foam cell formation and lesion growth. And KLF4 has really been one of the central transcription factors of importance in this mechanism. So to establish whether this is really true, whether Myatt really binds to this to the KLF4 promoter and drives transcriptional activity of KLF4, we used this luciferase reporter assay. We treated again human carotid artery smooth muscle cells with oxidized LDL, kind of like the epicenter of our cholesterol-rich Western diet. And using ox-LDL could see that the transcriptional activity in this KLF4 promoter region is really enhanced. And if we knock down Myatt, again, using gap-MERs, either with ox-LDL or without ox-LDL, we could really stop this transcriptional activity in this promoter region and kind of halt enhanced transcription of KLF4. And we could really pin it down to this nucleotide sequence that is around 100 nucleotides long. So if we mutate this binding site, again, KLF4 or the activity in the promoter region is really enhanced. And if we block this by using a mutated binding site, we can really stop transcription of KLF4. So what does this mean for smooth muscle cell phenotype and potentially also for the disease? So again, by treating using ox-LDL, treating these smooth muscle cells, we can really enhance Myatt expression and also KLF4 expression. And by limiting Myatt or inhibiting Myatt using gap-MERs, so site-specific antisense oligonucleotides, we can really not only block Myatt, but also KLF4. And if we look at different markers for smooth muscle cells and also macrophages or macrophage-like cells, so again, by using OXLDL, these smooth muscle cells, they kind of lose their smooth muscle cell phenotype. So they downregulate genes like ACTA2, which is shown here, but the same is true for others like smoothulin or myosin heavy chain 11. And they increase expression of genes that are more macrophage characteristic, like CD68 or the macrophage scavenger receptor 2 or galectin-3, so genes that are kind of prone to speak for a macrophage phenotype. And again, by blocking Myatt, we can kind of keep smooth muscle cells in their contractile phenotype and kind of block this transdifferentiation into a macrophage-like cells, at least in vitro. Myatt, however, not only plays an important role in smooth muscle cells, so we also looked into macrophages, in this case, actually from blood monocyte derived macrophages, that again, if we treat them with OXLDL, we see that Myatt is significantly increased, so expression of Myatt is significantly increased on the RNA level. And if we then, again, using this live cell imaging system, the InQSight system, where you can feed cells, in this case macrophages, with oxidized LDL, you can look at uptake of this, so more or less an assay of foam cell formation. And again, by blocking Myatt, we see that this OX-LDL uptake and foam cell formation is really hampered in these human blood monocyte-derived macrophages. Moving from in vitro into in vivo, so we used a couple of different mouse models. We were very fortunate that there is actually a Myatt knockout mouse that has been developed at the Riken Institute in Japan, and they shared it with us, and we crossbred that onto an ApoE background, so to get atherosclerosis-prone mice. And then used a couple of different models like carotid ligation and also this inducible plaque rupture model that is shown here, that we and others have used to kind of mimic in a mouse model a little bit closer what's going on in the human situation. So as many of you know, usually ApoE or LDL receptor knockout mice, they will really never develop very advanced lesions. They will never develop plaque ruptures, but with this more or less model that induces tensile stress on lesions, where you use this incomplete ligation of the common carotid artery and then put this conical cuff after 4 weeks just on top of this developing lesion, you can get ruptured lesions in around 50% of cases. And this is indicated kind of on these images to the right. So you can look at plaque formation. Use this in-house made cross-linked fibrin antibody, which is a very good indicator of an atherothrombotic event. And you can use SMA stains to really highlight fibrous cap formation, again, kind of being the last line of defense sitting on top of these necrotic cores in this mouse model. And so what we did with these knockout mice, or these double knockout mice, compared to the Myatt wild type ApoE knockout mice, so we're kind of able to show that these, based on plaque rupture and plaque advancement scores, that the double knockouts, so the Myatt ApoE knockouts, have a much more stable plaque phenotype. They're much more proliferative when it comes to the smooth muscle cells, and have significantly lower rupture rates, which again, we believe is mainly due to the impaired or very low expression of KLF4 in the double knockouts compared to the Myatt wild type animals. And then apart from using the mouse model, we're also very fortunate to have this large animal model in-house that we mainly use for translational studies and translational purposes. So it's a mini pig model, but if you look at the images, so don't be fooled by the term mini pig. They're quite big. They weigh between 90 and 100 kilos. And they're not only mini pigs, they also have the LDL receptor knocked out, which means that they spontaneously develop atherosclerosis. And if you put them on a high-fat diet, they do develop quite severe lesions and very advanced atherosclerosis, which is kind of shown on this right image. So this is, you can really tell this is the ostia going into the left coronary artery, and they do develop these very large, very advanced plaques that in this case have erosions and rupture. Actually some of these pigs, while being in an experiment, died from strokes and myocardial infarction. So again, this is of course not to replace any of the mouse models, but we use this a lot for translational purposes. As I said, So what we do see here as well is, especially in the pigs that homozygous and are put on a high-fat diet, so have very advanced lesions, that here we see Myatt and also KLF4, as well as 2 other target genes that I didn't talk about today, which are mainly responsible for this proliferative smooth muscle cell phenotype, which is ALK1 and EGR1. That all of them are also in this model significantly regulated, and there are studies now ongoing where we use drug-eluting balloons and mesh balloons and other devices to locally deliver non-coding RNAs, in this case Myatt, to advanced lesions to stabilize them and to block or limit the risk of acute rupture in this preclinical model. And that's, so this is kind of like the last slide already, kind of going full circle back to the human material. So we've also started, of course, using single cell sequencing of our plaques. And we've been trying to, in particular, look into plaque specimen that we get out of our surgery suites, where we not only have very advanced lesions, but also get an adjacent piece of tissue that is usually, it's still disease, but to a much lower extent disease, so kind of like an internal control, and then perform single cell sequencing of these advanced plaques and compare them to these controls. And of course, there's again then the opportunity here to look at specific long non-coding RNAs that are specifically expressed in certain clusters. And usually for all of these clusters, so these are not the real names. Again, this is very early and very preliminary. But there is, of course, usually most of the top targets, at least in our hands, are long non-coding RNAs or are non-coding transcripts that seem to be very important in regulating functions of these different cell clusters. And maybe next time I can present the seminar, I will be happy to more talk about this study and some of the work that has been going on using this very valuable tissue material. Okay, so in summary, I hope I was able to convince you that long non-coding RNAs are very important regulators in lesion vulnerability. In particular, this long non-coding RNA Myatt that I talked about affects proliferation and survival of vascular smooth muscle cells. So proliferation really mainly through these 2 transcription factors, EGR1 and ALK1, that I didn't talk about too much, but it's a little bit complex to talk about this in 30 minutes, as well as to talk about the KLF4 part. And again, this effect on enhancing, so triggering transcriptional activity in the promoter of KLF4 and really driving this pro-phagocytotic phenotype in the smooth muscle cells via KLF4. And then lastly, so of course, we believe that therapeutic targeting of this long non-coding RNA might really enhance lesion stability, and that's something we're currently testing in our large animal model. And with this, I would just like to thank the people in my Munich lab, our collaborators from vascular surgery in Munich, very importantly also the lab in Stockholm that we still have, and people like Ulf Edin and Lubica Matic from vascular surgery. Again, there's very close collaboration with Bike and the people there, collaborators from other institutions, all of our funding agencies. Many thanks to you for listening, and I'll be happy to answer any questions. Thank you.
Okay, great. Thanks, Lars. So you may not have been aware while you were presenting, we had hackers join our meeting and completely take over our chat function. So I don't know if we've disabled it. We've tried. So I think, no, not yet. Okay, so the chat is not the way to do this. We're going to instead, everybody, if you click on the participants at the bottom, if you, there's a raise hand function. If you go to your own, what do you call it? Your own profile, you can say raise hand in the participant section. So if you have a question, please raise your hand and we will try to answer your question. Okay.
George Shakir.
Yeah, did you hear me?
Yes.
Yes.
Okay. Yeah, thanks for this presentation. I want to ask like a couple of questions. It's quite small. So how much the homology between between the 3 animals, or the humans and mouse and the other animal you used, and what would be the normal function of this long non-coding RNA in other organs or Like what does it do? And also, do you have an idea, or you think like how the expression of this long non-coding RNA is affected by the LDL? Like, what's the sensing mechanism? Like, does it overexpress with the huge amount of LDL? Does it down express? And what are the linkers between like cholesterol coming out from the receptor, from the outside the cell to inside, and then enhance the expression of this long non-coding RNA?
Okay, so that's a lot of questions. I try to dissect them. So initially, Myatt was initially transcribed. So now I have a visitor. Myatt was initially transcribed as was initially a GWAS hit, actually, in a Japanese cohort looking at risk of myocardial infarction. And it was detected there. So it's that's kind of where it has its name from, but its biological function has really never been really studied by many groups. It's not solely expressed in the cardiovascular system. So there's it's also a GWAS hit, I think, in epilepsy and some other neurogenic disorders, and that's actually where the group that had this mouse at the Riken Institute, what they were studying was actually epilepsy and other psychiatric disorders. So it's not exclusive to the cardiovascular system. It's really pretty much expressed in every cell, but it's for a long non-coding RNA, it's fairly enriched in the cardiovascular system. It's fairly high expressed, has fairly high expression levels in smooth muscle cells, also in macrophages, endothelial cells, cardiomyocytes. So it's almost in every cell. Then so the other questions, so the direct mechanism of how OX-LDL triggers Myatt expression, we really don't know. So that's something we have not really looked into, to be honest. And then again, the main downstream function. So we've done experiments using these reporter assays and also RNA immunoprecipitation IP. So it's really not interacting with a lot of genes. Mainly again KLF4 and other transcription factors, and these other transcription factors, there's now studies coming out that also show that it's important in cancer disease. So this mechanism of Myatt targeting EGR1, so early growth response 1, which is an important tumor suppressor gene, I think, has been studied or has been shown in other disease models as well. I'm not sure if I captured all questions.
The last one, like, is it like is it homologous between the humans and the mice and this kind of small, not small pigs?
Yeah. So between humans and pigs, it is. In mice, it's there, but it's somewhere else. I don't remember the exact location. So it's conserved, but it's not synthetic.
Thanks.
Okay. Sorry, I'm just looking here. Ebru, can you take over for a second? You're on mute.
So Chiara should be unmuted.
Yes.
Okay.
Yes.
Yes, I'm here. Hi, everyone. Lars, very nice presentation. Thank you so much. I just had a few, questions for you. I guess the first one is about the cell specificity for Myatt. So you mentioned that is expressed based on your analysis by alpha-actin positive cells, but not by CD68 positive cells. And I guess that was the rationale for using a full knockout in your mouse experiments. But, are you concerned about the expression of Myatt and potential, unanticipated effect that are unrelated to, I guess, vascular smooth muscle cells expression in the context of the plaque?
Yeah. So a great question, of course. So it's again, it's not exclusive to smooth muscle cells. Again, we try to, at least based on the human expression profile, try to nail it down as close as we could to being important in smooth muscle cell morphology. But again, it's also expressed and of importance in macrophages, where it seems to trigger actually NF-kappa B translocation. So as I've shown, I mean, we've done some experiments in macrophages, so it's definitely important there as well. If we now look at our single cell sequencing data that we have, again, this is based on 10x genomics, so we only have around 1,000 genes per cell. And there it was really not so much in smooth muscle cells, to be honest. It was actually more in macrophages there. So it's which is why I didn't show any Myatt data relating to the single cell sequencing.
Well, but that could be, you know, I'm assuming you might not have a full spectrum of disease, so you might just capture a different state.
Of course.
The other question I had, it was about some, you presented some knockout data from, I guess, human vascular smooth muscle cells. And I saw there, and maybe it's just that picture, that it looked to me that you had more cells in the knockout experiment as opposed to the wild type, but then you show that there, so I was thinking, oh, maybe there is some effect on proliferation, but then you show in the mouse that is actually the opposite, and you have more proliferation in the wild type, right? If I remember correctly. So
no, so it's the same. So the knockdown in smooth muscle, in human smooth muscle cells, triggers proliferation, triggers survival, so less apoptosis, and the same is in the mouse. The mouse also has it's more proliferative and has a more stable plaque phenotype, so more stable fibrous caps. So that's actually consistent between the two. Sorry for not being clear.
So you answered my question. I might have missed that. Thank you.
Yeah, sure.
Thank you, Chiara and Lars. So Roberta is next. Roberta Gottlieb.
Hi, I really enjoyed that. This is a little outside my comfort or knowledge space, but I was wondering, given the expression in endothelial cells and smooth muscle, whether or not you see an upregulation in hypertension.
Yeah, so that's also a very good question. I mean, we know that in hypertension, smooth muscle cells also start to proliferate, and there's mechanisms of remodeling involved. So for sure, there should be a deregulation in this. Based on our data, again, I mean, I think what Chiara just said is very important. So we just have a snapshot of disease. So this is very advanced lesion development in humans. And I think it would probably be the same in advanced stages of patients with hypertension. What kind of happens during, I don't know, plaque development, atherogenesis, and remodeling in hypertension might be very different to what we see with this one snapshot of very advanced lesion development. So, but I'm sure, so it would definitely make sense if this is at least deregulated in the vasculature in patients with hypertension.
Okay, this is Lale. We will stop the questions. I know Catherine also has a question, but let's save it for later. We need to move on. Our next speaker, we thank Lars, by the way. Our next speaker is Esther Lutgens. Esther is a professor of vascular immunopathology at Amsterdam University Medical Center. She works on the modulation of co-stimulatory pathways and the immune system and vascular inflammation. Today, she will tell us about her work on immune checkpoints in atherosclerosis with a focus on GITR. Esther, can you share your...
Yeah, I'm trying. Do you see my screen?
Yes.
Yeah. And sorry, Esther, I said that you were in Germany before and I forgot that.
I actually also, like Lars, have a dual appointment, but these days with the coronavirus, I'm stuck in the Netherlands. I'm not allowed to travel to Germany anymore. So it's mostly Zoom-based affiliation with the LMU now. So, but thank you for your introduction, Lale. Thank you for staying around and listening. And today I'm going to tell you some of our new data, something on immune checkpoints in atherosclerosis, and a new molecule we fell in love with, and it's called GITR. So I'm also investigating atherosclerosis, just Lars, and atherosclerosis, as you all know, is the underlying cause of most of the cardiovascular diseases. If you take a close look at the plaque, you can see that an atherosclerotic plaque can have many phases. This is like a non-diseased artery. This is an artery with an atherosclerotic lesion, stable lesion, thick fibrous cap, clinically non-significant. And this is an atherosclerotic lesion that has encroached the lumen, has underwent plaque rupture, and this is clinical symptomatic disease. Atherosclerosis is a lipid-driven immune disease, so not only cholesterol, triglycerides, and lipids like Lp(a) are involved, but also the immune system. And atherosclerosis is also a very interesting disease. It's not boring, but all people develop atherosclerosis differently. Some people have more lipid problems, so they have high cholesterol, and other people have more of an inflammatory component of atherosclerosis. So here you can see the inflammatory component. This is pure plaque rupture. This is encroaching of the lumen. This is more lipid-driven atherosclerosis with a huge lipid-filled core. So what my lab is interested in is why atherosclerosis has so many phases and how immune cells especially regulate plaque phenotype. And already Kandinsky, whom I stole this picture from, appreciated the beauty of atherosclerosis. So currently we have lipid-lowering therapies, but since 2017, we also know that immunotherapy is very favorable for treating atherosclerosis. However, we're just at the beginning of designing immunotherapeutics for atherosclerotic disease. The CANTOS study has shown that if you block IL-1 beta with an anti-IL-1 beta antibody, you get a reduction in primary and secondary cardiovascular disease endpoints. However, there's no change of overall mortality, and there's a little increase in the risk of infection. So I think we and all of the people here can do much better. And I think we need to invest a lot of resources and a lot of efforts in our research on how inflammation and the immune system affects atherosclerotic disease. So I drafted a little picture here with future immunotherapy. So there are several classes of inflammatory agents that have the potential to once become immunotherapeutics. And one of them is the immune checkpoints. And this is the personal hobby of me and my lab. And what we would like to accomplish is that we not only want to reduce primary and secondary cardiovascular disease endpoints, but we also would like to accomplish a cardiovascular disease mortality decrease, and with most probably, and we hope, without immune suppressive side effects. Let me see. Okay. So what are immune checkpoints? So immune checkpoints, if you look into your classical immunological textbook, they are divided in co-stimulatory and co-inhibitory molecules, and they are essentially painted as the interaction between an antigen-presenting cell and a T cell. So you have three signals. Signal 1 is the interaction with the T cell receptor and the antigen MHC class 2. Signal 2 is actually required to get a proper immunological reaction. This is the checkpoint signal or agonistic checkpoint signal, or also called co-stimulatory signal. That's really needed to propagate an immune response. And signal 3 is the help of several cytokines and chemokines. So you need all three of them to accomplish a proper immune response. But in the past years, it also became clear that, that these immune checkpoint molecules, that they are not only expressed on antigen-presenting cells and T cells, but they are actually present on all kinds of cell types. So smooth muscle cells, endothelial cells, even epithelial cells, several cancer cells. And immune checkpoints were found to regulate cell-cell communication within the immune system, and not only restricted to APCs and T cells. You have several classes of immune checkpoints. One of them is the co-stimulatory molecules. You also have co-inhibitory molecules, and inhibiting those are very popular in current oncology treatment. These are the inhibitory immune checkpoints, as you call it. There are also some smaller classes, and there is an ever-growing group of other immune checkpoints. Today, I will particularly focus on co-stimulation. So immune checkpoints warrant proper immune cell communication, and also inside the atherosclerotic plaque. If you see if you take a closer look at the atherosclerotic lesion, you just don't see only macrophages or only T cells or only lipids. You see lots of cells. You see macrophages, T cells, B cells, monocytes, some dendritic cells, even some B cells, and not only inside the plaque, also in the lymphoid organs outside of the plaque. And in order to drive disease or hamper disease, all these cells need to talk to each other, and they do that with the help of immune checkpoints. My laboratory thinks that these immune checkpoints are quite good therapeutic targets, and we have been working for decades on one of these immune checkpoints, and that's CD40 ligand and CD40. I'm not going to talk about that much today, but I will just use it as an illustration how powerful these immune checkpoints can be in cardiovascular disease, and also to show you that they can be targeted safely and are very efficient in therapy. So we picked up CD40 ligand in a microarray experiment where we compared stable atherosclerotic plaques versus ruptured atherosclerotic plaque, and we found CD40 ligand to be expressed in ruptured atherosclerotic lesions. We also found its counterpart, CD40, to be expressed, and what we found is if you block CD40 ligand or CD40, you get very small atherosclerotic lesions, which also had a very favorable stable plaque phenotype. So these lesions were very poor in inflammation, very high in fibrosis, and this is kind of the lesion you want to get in patients. However, if you want to use it as a therapy, if you would block CD40 totally or entirely, you would get immune suppression, because CD40 ligand, CD40 interactions are obviously very important in the immune system. So we came up with an alternative strategy. So CD40 exerts a lot of different signaling pathways. And in macrophages, CD40 signaling goes via TNF receptor associated factor 6. And these TREFs are also not unique to CD40 signaling, but they're also involved in other signaling pathways. So we were able to design a small molecule inhibitor that specifically could target CD40 TREF6 interactions. Thereby, we could block macrophage activation and atherosclerosis. And we were able to keep a normal immune system because the CD40 B cell function, for example, went via CD40 TREF2, 3, and 5, and that remains still intact. So we could show that in several of our papers. Here is the TREF6 molecule. Here is a small molecule inhibitor that targets the CD40 binding groove in the TREF6 molecule. And when we give this molecule to mice, we could even reduce established atherosclerosis. So we aged our mice until the age of 22 weeks, treated the mice for 8 weeks or 6 weeks, and then we could see that atherosclerosis was reduced. These are the mechanisms. So if you would block an immune checkpoint, an agonistic immune checkpoint like we do, you have to be quite careful. You have to kind of go into the signaling pathway. Here we blocked it, the CD40 TREF6 signaling pathway. We reduced monocyte and macrophage recruitment and activation, blocked atherosclerosis, whereas we kept the normal CD40 mediated immune reactions intact. We've now done a non-human primate study with these compounds. We put it into nanoparticles, and what we could see is that the distribution pattern was quite okay, and we had non-toxicity. So these compounds are really safe, no immune suppression, and we hope to come up with a compound that's we can put into the clinics, but that's some years away. So until now, there are several groups that propose that immune checkpoint therapy is very valuable in atherosclerosis. Our group is one of them, is our CD40 TREF6 inhibitor. There are also people who develop antibodies against CD40 or CD40 ligand, and there's also a group that's trying to develop anti-CD86 antibodies. But this is all work in progress, and it will take some years before this is being employed as immunotherapy. However, as I learned in the past years, there's more than CD40 and CD40 ligand, although you can investigate, I think, 50 years of your life studying these two molecules, but there are more immune checkpoints and they all interact with each other. And one of these immune checkpoints is GITR. And GITR is not that well known. GITR stands for glucocorticoid-induced TNF-related protein, but has nothing to do with glucocorticoids. It's just the name. It was first found in glucocorticoid-stimulated cells. The ligand of GITR is GITR ligand, and we stumbled upon GITR and we would like to investigate it because GITR was long known as a regulatory T-cell marker. However, this has already been abandoned. It is a regulatory T-cell marker, but GITR is also expressed on effector T cells, also on antigen-presenting cells, macrophages, mast cells, also in endothelial cells. And the role of GITR was quite diverse. In some disease models, it has an active driving role in inflammation, and in other disease models, it really plays a regulatory role in disease models. So we thought, well, what role would GITR play in atherosclerosis? So we also made use of a carotid plaque biobank, and we used the biobank from Lund University together with Isabel Gonçalves and Jan Nielsen, and this is the carotid plaque imaging project. So we also have these beautiful endarterectomies, just like Lars shows, they look the same. This biobank has now about 1,000 plaques, and it's also coupled to patient data. So we divided the plaques based on the patient data. So we had patients with cerebrovascular disease and patients without cerebrovascular disease. And what we wanted to do is to look at GITR. Is GITR actually expressed in human atherosclerotic lesions or is it not? My postdoc, Annelies Chamy, really spent about 3 to 4 months in Lund staining GITR, and she found that GITR in human atherosclerotic plaques is present in T cells, as would be expected, but was even more expressed in the macrophage. We use several markers, CD68 is one of them. It was also expressed in the endothelium and very little in smooth muscle cells. We also wanted to know, is its expression correlated to cerebrovascular disease? And indeed it was. In symptomatic patients, if you have plaques from symptomatic patients, GITR expression was elevated and significantly higher than its expression in asymptomatic patients. If you have such a database, you can also correlate GITR expression with all kind of other stainings and with all other parameters that are available in the database, because it takes a lot of years to build such a database. And what we could see is that GITR was correlated, positively correlated with parameters of plaque vulnerability, like oil red oak content was negatively correlated with alpha smooth muscle cell content. It was positively correlated with lipid core content, etc. So we thought, well, it might be an interesting molecule to investigate. We also took plasma data from another cohort. So this is not from the biobank, but this is from the summit cohort. And there we could compare soluble GITR levels in the plasma of healthy people versus people suffering from cardiovascular disease. And people with cardiovascular disease indeed have elevated soluble GITR levels in their plasma. So then we thought, well, it's there, it's expressed in immune cells, but what does it do? So we were lucky to get a hold of the GITR deficient mouse model. We backcrossed them to ApoE knockout mice. We gave them a diet and then start to investigate them. First, we checked whether GITR in mice had indeed the similar expression pattern than in humans, and this was indeed the case. Some of the GITR is expressed in T cells. Most of the GITR is expressed in the macrophages in the lesion, few in smooth muscle cells, and also a very nice endothelial lining. And the special part was that GITR was especially expressed in the endothelium overlying the atherosclerotic lesion, as you see here, and it's negative in the other one. So what happens to atherosclerosis? So if you don't have GITR, you get a reduction in atherosclerotic lesions. The reduction was significant. It was significant in males, females, and in 2 different locations, but it's not like a 70% difference, it's about 30%. But the most striking effect there is that there was a reduced influx in macrophages. So reduced macrophage content and also a reduced necrotic core content, reduction in necrosis if you don't have GITR, and we made a plaque vulnerability index, and we also could see that plaque vulnerability in GITR deficient animals is lower. We were actually quite surprised that we saw such an effect on macrophage content. We also looked at lymphoid cell content, T cell content, CD4, CD8 positive T cell content in the plaque, but that was not altered. And also if we had effects on these animals, because GITR is particularly known to have a function in autoimmune disease and T cells, we really could not see any difference in T cell numbers, T cell activation, not in the CD4, CD8 content. So we were really triggered towards what's the role of GITR in myeloid cells. GITR was also very nicely expressed in the endothelial lining overlying atherosclerotic plaques. So we thought, well, maybe it's also involved in endothelial cell activation. And we did many stainings and also many leukocyte adhesion assays, and it's actually not. So the conclusion is that deficiency of GITR does not affect endothelial cell activation. And I here show you staining for ICAM and VCAM. Then to really prove that it's the hematopoietic GITR that is really key for the reduction in monocyte and macrophage recruitment, we did a bone marrow transplantation, and here we confirmed that if you have GITR deficient bone marrow, you have a strong reduction in macrophage content in your atherosclerotic plaques. So there is not so much known about the function of GITR in monocyte and macrophages. So what we did is we took classical and non-classical monocytes from the spleen, and then we did RNA sequencing on these monocytes. And I here show you the results of the non-classical monocytes. And what we actually found is that there were metabolic alterations in these monocytes, and also pathways of recruitment were altered and were decreased. So there were less reactive oxygen species, less mitochondrial function, and we thought that the activation status of the monocyte must be altered. We also wanted to do a recruitment assay, and we once more wanted to prove that it's really the myeloid GITR that's responsible for the plaque phenotype and not the GITR on the endothelial cells. So what we did is we either took a carotid artery of a GITR positive mouse or a GITR deficient mouse. We mounted that between 2 glass pipettes, and then we perfused GITR positive or GITR negative monocytes. They are labeled green and red, and then we counted them while we perfused these carotid arteries, and these are 2 photon images. And if you have GITR on your endothelium, there is a reduction in leukocyte adhesion from the GITR negative monocytes, and the same is true if you take GITR deficient endothelium, so you have the same results. So GITR deficient endothelium does not play a role. We also looked at some monocyte markers, and we also could see that they are reduced in absence of GITR. Let me see. We also looked into the macrophage and GITR. So here we actually saw the same pattern as in the monocytes, but we also saw a huge reduction in migratory pathways. And this is also what we could confirm. I missed a slide, I think, but let's move to the conclusions. So we concluded that if you have a low GITR content, content, the plaques are mostly asymptomatic patients. If there's no GITR in your plasma, this also reflects a more healthy cardiovascular system. In symptomatic patients, we found more GITR, and we could also find that if you don't have GITR in the knockout mouse, you have a reduction in leukocyte recruitment, also a reduction in monocyte activation, macrophage activation, and you have a smaller lesion formation, and they have also more stable lesions. So this is like the summary of the GITR story. We're still working on it a little bit, but there are of course many more co-stimulatory and co-inhibitory molecules we would like to investigate. And what we are currently doing, and this is really unpublished, and I just wanted to share it with you. I think it's very important to see which immune checkpoint is expressed on which cell type during the different stages of atherogenesis. And in collaboration with Claudia Monaco in Oxford, we are doing a huge cytoscreen. So here you see lots of co-stimulatory and co-inhibitory molecules. You see the aorta, and we are actually trying to build an immune checkpoint atlas of atherosclerosis. But as I told you, it's still work in progress, and I hope that with this virus pandemic, that I hope in a year that we have a very nice overview of which immune checkpoint is important on which cell type and when in the disease it becomes important, so we can better develop our therapies. I hope I have convinced you that immune checkpoints are important in atherogenesis. I hope I've also shown you that immune checkpoint signaling can be targeted efficiently and safely without immune suppressive side effects. But I do think the immune checkpoint interactome still needs to be unraveled. And with our lab, you will hear some more presentations, I think, on novel, powerful, druggable candidates such as GITR, CBL-B, and another one is VISTA. So I haven't done this work all by myself. So this is the group with also the Munich group with Christian Weber and Dorothee Otzler. These are all the people I have to thank, and they have all collaborated to these projects. And thank you all for sticking around, and I'm happy to answer some of your questions.
Hey, great. Thank you, Esther. It was a great talk. If anyone has a question, please raise your hand. I see. Yeah, I am unmuting you right now. It's Ada, I guess, right?
Catherine. Catherine Ho.
I think Catherine's talk question was for Lars. Maybe if we have time, we'll take her question. But let's go with Ada.
Can you hear me?
Yes.
Yeah.
Okay. Thank you for the beautiful talk. I was wondering if, because you showed in the GITR mice that were basically no effects on T cells. So I was wondering, what is GITR doing in macrophages and is not affecting T cells if it's a co-stimulatory molecule?
So we now know that many of these co-stimulatory molecules that have been always assigned to T cells play actually a very important role in the macrophage. Like CD40, for example, plays a very huge role in the macrophage and the ligand in the T cell. But the GITR is expressed both on the T cells and the macrophages. And I don't say that it doesn't play a role, but I think in atherosclerotic disease, GITR plays a very important role in the macrophage as well. In previous times, I think about 6 years ago, we also made use of another model for GITR, where we specifically activated GITR in the T cell by having GITR ligand overexpressed in a B cell. And there we actually saw that in atherosclerosis, this T cell actually had a more protective effect. But if you block it, then and this was a very unphysiological model. If you look in a more physiological setting, in atherosclerosis, it seems that GITR in the macrophage is actually more important in this chronic inflammatory disease than GITR in the T cell. So
do you know what it's doing in the macrophages that is unrelated to T cells?
Yeah, we're still kind of puzzling. If we stimulate the macrophages with the GITR ligand, with the DTA, we see that they become pro-migratory. We haven't figured out the signaling intermediates yet. We're planning to do that. We know in the T cell that they signal via TREF2. In the macrophages, we're not so sure. But we see that with many co-stimulatory molecules that they are expressed on a lot of cell types, and that all these cell types also kind of exert their own signaling pathway with their own adapter molecules and own functions. So we know that it's important, but how exactly, I think we need to dive into that a bit more.
Okay, so now let's have Catherine ask her question. I don't know whether, Catherine, you're unmuted now, whether your question was for Lars or Esther. Please go ahead. Sorry.
Great, thanks. Can you hear me okay?
Yeah, I hear
you. Yes, hi. Lars, are you still on? This was actually a question for you, although Dr. Litkins, it was a fantastic project and overview. I'm impressed. So is it okay to go back to ask Lars a question though?
Yeah.
Okay. My question centers on the therapeutic aspect that you were trying with, I assume you're probably Yucatan pig model. Do you have a sense of whether, you know, a single administration of the drug-eluting balloon, or at least layering on some form of probably transient knockdown of Myatt, whether or not that has lasting effects? Meaning, you know, many of us are waiting until there's a symptomatic lesion or a progressive lesion before we actually intervene. So do we have an opportunity to actually change the trajectory at that point? And is it lasting?
So I mean, great question. Hi, Catherine.
Hi.
So it's we don't know yet. So again, I mean, the idea is we've mainly been working on aneurysm models with different ASOs and local targeting. The big advantage, of course, is that with this local targeting, you can really get a lot of drug compound into the lesion. We're a little bit worried right now, and that's what we've seen as well, that with some of the local delivery tools we've been using, we kind of see some friction on the lesion, and that of course might further unstabilize it. So the idea with the ASOs, at least with the ones we've been testing there, that they have a very, I mean, they're very stable, so they work up to 28 days. So at least so we fluorescently tag them, we can see where they go into smooth muscle cells, and we can image them for up to 28 days. They're quite long around. The idea from a more clinical perspective, of course, would be to kind of combine this with imaging. So really detect really vulnerable lesions and have a tool for patients that are either unsuited for surgery or something else, that we can use this really to acutely stabilize something. That's the main clinical application, I guess.
Okay.
Right. Great. Next one is Katie. Katie, please go ahead.
Okay. Oh wait, hang on. I think I'm muted. Am I unmuted now? Okay. So this is a question for Esther. I'm wondering, and maybe I missed it in your talk, but does GITR have only the one ligand, like the GITR ligand? And if so, do you know its reciprocal expression, like is it only or primarily functioning in monocytes or macrophages, or yeah?
Yeah, it's not known whether GITR has more than one ligand. Maybe it has, but it has not been discovered yet. GITR ligand is one of the ligands of GITR, but it's also quite promiscuously expressed. So it's also sometimes on the same cell type, you have the ligand and the receptor expressed. So GITR ligand can be also expressed on the macrophage, can definitely be expressed on the endothelial, but GITR is also on the endothelial. So from the mouse models and the transgenic mouse models, it's pretty clear if you have a GITR ligand overexpression, you get a huge stimulation of GITR. So that interaction really occurs. Whether there are more interactions still needs to be investigated. So.
Okay, great. And I guess we have one more question. It's from Sipka. You're muted now. Please go ahead with your question.
Thank you. So I have a question for Esther. Thank you for a nice talk. So in your talk, you mentioned that like CD40 ligand has different signaling cascades in immune cells and in macrophages. Like in macrophages, it's via TRAF6, and in other immune cells, it's via TRAF2, 3, and 5. So do you have any insights about the GITR signaling, why it's behaving differently if it's knocked out in the myeloid cells and in the lymphoid cells? Does it also have a different signaling cascade over there?
It's a very good question, and we're still figuring it out. So this was like the first study, but we will certainly dive deeper into that. From literature, we know that GITR has a favor for TRAF2, but we do not know if this is specific for the macrophages. We need to sort that out. So it's not known yet. So.
Thanks.
Okay, I guess that's the end of the questions. So thank you, everyone. Thanks for the speakers. Thanks to the speakers, Lars and Esther, amazing talks. Next week, we have, again, two amazing speakers, Katie Rayner and Ed Fisher. And it's going to be at the same time. Perhaps we'll try to take more actions to prevent the people from, our Zoom. Okay, so thanks everyone. We'll see you at noon again with the same Zoom number. Bye now.
Okay. Good. Thank you, everyone. They all. I don't hear you.
So I will end the meeting and the organizer can rejoin.
Yeah.
Okay.